Hello and welcome to Nikolai's genetics lessons, and today's problem is which of the following sequences is most likely to be cut by a restriction enzyme with the following recognition sequence. Two sequences are template and non-template strand. So here is the template strand and this part represents non-template strand. So first of all, we are looking for sequences that are going to be palindromic. What we mean under the palindromic, take a look. For example, this is going to be example what we call palindromic sentences, or it can be words in English. For example, don't not, we can read both ways, don't not, and backwards we can also read it the same, and the meaning is going to be the same, don't not. And all the sentences we can read forward and backward, and the meaning is going to be the same. But in genetics, this is not quite the same, what we call palindromic. For example, let's say we have a sequence which is going to be 5' end A, T, A, and 3' end. If you think that this is palindromic sequence, because we can read it both ways, and it's going to be the same, it's not actually quite so, because we have to build another complementary strand of the DNA. And this time 5' end is going to be here, adenine base pair with thymine, so thymine here, adenine, thymine, and 3' end here. So as you see, top strand is not the same as bottom strand, because sequence here is 5' end AAT, AA, and at the bottom strand, complementary strand, the sequence is 5' end TT, ATT, 3' end. So this is not example of the palindromic sequence. And out of these four variants, we have one sequence that is palindromic, and this is sequence D. Let me write it down one more time. So we have 5' end, and then we have A, T, T, and 3' end. And complementary strand of the double-stranded DNA is going to be 5' end here, then A, T, and 3' end. Now let's check if this is going to be palindromic sequence or not. 5' end AAT, 3' end. And complementary 5' end AAT, 3' end. We call such sequence palindromic because we read it the same on one strand of the DNA, and the same sequence is going to be on the other strand of the DNA. One goes forward, another goes backward. And also we are looking for the sequence according to our problem, which is going to be 5' end AT, 3' end. And this is going to be on the top strand AT, this sequence, and on the bottom strand it's going to be AT, this sequence. So this is going to be a recognition site for restriction enzyme. And as you see, the correct answer is answer D. And here's our sequence AT, and here's AT. And this is all for today. Subscribe and see you in the next video. Goodbye.